Hello and welcome everyone to this final podcast on this section on chromosomal rearrangements and changes in copy numbers. And in this podcast, what we're going to talk about are some of the mechanisms that cause aneuploids and polyploids. This first mechanism, and really it's the main mechanism we want to talk about, is called Nondisjunction. And really what happens with nondisjunction is you can have homologous chromosomes that are supposed to, well, first of all, remember they synapse during prophase 1, and then they line up during metaphase 1 where that metaphase plate is in between them. And they're supposed to pull apart, but every now and then they kind of get stuck together because the proteins that hold them together aren't degraded, so they're stuck together. And then when it divides, both of these go towards one cell. You can also have a situation where during meiosis 2 or during mitosis, the sister chromatids, as they line up along that metaphase plate, they're supposed to pull apart, but sometimes with nondisjunction, they stick together. And instead of going in this direction and this direction, the two sister chromatids, they both go in this direction. So I'm going to write under here, on this side here, this is what we would see in meiosis, or mitosis, meiosis 2 I should say, or mitosis. And this one is meiosis 1. Now I want to talk a little bit more about nondisjunction in meiosis 1 and meiosis 2 and see what kind of gametes are produced. First I want to talk about nondisjunction in meiosis 2. So in this case we're going to start off with a cell, and I'm only going to draw one homologous pair here. And remember, they attach to each other during prophase 1, and then during metaphase 1 they line up along this metaphase plate, and then they pull apart from each other in anaphase 1 and telophase 1. However, sometimes these two chromosomes stick. Well, actually I shouldn't say sometimes. They always stick with each other during synapse, which allows that crossing over to occur. And during metaphase 1 and anaphase 1, what's holding them together is supposed to come off. But sometimes that doesn't happen. And so this is going to be nondisjunction in meiosis 1. So during meiosis 1 here, we form two cells. In this cell, we end up with both copies of this chromosome because it went through nondisjunction, they didn't separate. And over here we get no copies. Now then, as we go through meiosis 2, this cell will form two new cells. And what happens here is these two sister chromatids come over here and then they do separate. And these two sister chromatids come over here and they separate. So we end up with two copies of each chromosome in this gamete. Over here, this cell will divide twice, also in M2, and it gets no copies of that chromosome. Now, let's say these are sperm, and if this sperm is to fertilize an egg that is a haploid, as most eggs are, then you're going to end up with a cell that has these two chromosomes, that is, that fertilized zygote is going to have these two chromosomes from the sperm and the one chromosome from the egg. So ultimately, this is going to lead to a trisomy, as will this. And down here it's going to lead to a monosomy, because there's no chromosome, this blue chromosome doesn't exist in the sperm, and there's one copy that's in the egg, so when fertilization occurs, you just have one copy. So this is monosomy, or it will form a zygote that is a monosomy. So this is what happens during the phenomenon of nondisjunction during meiosis. Oh, I just noticed kind of a big mistake here. This is meiosis 1, not 2, meiosis 1. Nondisjunction during meiosis 1. My apologies for that up there. Now let's look at what happens with nondisjunction during meiosis 2. And now I'm going to change this back to 2, because now we're looking at nondisjunction in meiosis 2. In this scenario, the two homologous chromosomes separate. Meiosis 1 goes off with no problems at all. So we end up with our two homologous chromosomes separated from each other, and each of these developing gametes, future gametes, have 2 copies of each of the chromosomes in them, as they should. Now when non-disjunction occurs, it would be incredibly rare for it to happen in both. So with this one here, let's just say that this bottom one has no problems at all. The sister chromatids separate just like they should, and we end up with 1 chromosome in each. I'm going to erase this over here too, because these are going to change. Now, again, non-disjunction in meiosis 2, these 2 sister chromatids fail to come apart. And so now what we end up with is one of the cells like this, with 2 copies of this chromosome in, and this cell with none. Now just like we said before, when this egg, or sperm I should say, fertilizes an egg, it will produce a progeny that has 2 copies of this chromosome, because the egg also has 1. And so this one will survive, will be a euploid. Same with this one, 1 copy from the sperm, 1 copy from the egg that we don't see here, but it's there somewhere. It's also a euploid. Here, what we see is there's no copies from the sperm. There will be 1 copy from the egg, and so we're going to end up as a monosomy with this one. Here we have 2 copies from the sperm. It'll get 1 copy from the egg, so then that's going to be a trisomy. Again, this gamete isn't a trisomy, but once it fertilizes the egg, that is a euploid, then it will form the trisomy. So it's important to recognize that non-disjunction can occur at either of these 2 stages of meiosis, meiosis 1 or meiosis 2. And I'm going to talk about this really interesting phenomenon called Mosaicism. Individuals who have a mosaic phenotype, or this trait, is a good explanation that sometimes non-disjunction occurs in mitotic cells. And so if we have our zygote here, and then it divides several times, maybe many times, right? And we end up with lots of cells here, all doing fine. But this one here, or these 2 here, they form some sort of aneuplooid. Let's just say it's going to be a trisomy 18. Usually individuals who are trisomy 18, if they survive embryonic development, they don't live for much more than 2 to 5 years. And that would be in the case where the aneuplooid, or the non-disjunction, occurred in meiosis 1 or 2, so that every single cell was a trisomy 18. But as you can see, as I drew here, that only a small portion of this growing zygote, or this growing embryo, has this trisomy 18. As this individual grows, and this is going to be not a great drawing, so I'm just going to stick with my stick figures here. In general, most of their cells are going to be of, say, this green type. And the green here is, why don't we just put it here, these are euploid. They have 2 copies of every chromosome, so they're perfectly healthy. And this individual that forms is also going to be pretty healthy, or more healthy than its counterpart, I guess I should say. But what I'm going to do is I'm just going to make some random red dots on this one. Some of its tissue, some of the cells in it, will have an aneuploid associated with them. It makes their phenotype of trisomy 18 less severe, because not every single cell in their body is this trisomy 18. This mosaic phenotype can also help explain why individuals may have, say, like a trisomy at chromosome 4 or 5, which is always lethal if it occurs in the gametes before they fertilize. But you could have a mosaic individual survive with that if it's only in a few of their cells. So that's what a mosaic is. Okay, I want to talk about 2 more terms. The first one is an autopolyploid. This refers to an increase, so up arrow, increase in chromosome sets in a single organism. And so this would be our essentially our polyploids, but it's a unique one in the fact that it's only within the same organism. And we could get this if, for instance, let's draw a big cell here, and we'll draw this metaphase plate. And let's say along this metaphase plate we have several homologous chromosomes. This is going to be meiosis 1, say. So I'll just draw 3 here. They're all long here. And remember, it's these spindle poles that are supposed to hold them apart. But what if there was a general defect in these spindle poles, and they didn't function. And as a result, they didn't attach to the poles. They didn't attach to the centromeres. So let's just remove them. In this case here, you could end up with a cell that has all of these in there. You may end up with a cell that looks just like this, particularly after it goes through meiosis 2, right? That has 4 copies of the blue one. It's going to have the light blue one, then 4 copies of the dark blue one, and then 4 copies of the green one. So this would be an example of how you go from a diploid to a tetraploid by losing all control of the spindle apparatus. You could also imagine some other scenarios where maybe in meiosis there was non-disjunction at every single chromosome. Again, that could happen. And we already mentioned before how these polyploids typically are sterile. Okay, I want to talk about one more term here, and this other term is called allopolyploid. And this is a polyploid organism that contains more than one chromosome set, but they come from different species. Polyploid that's made using chromosomes from other species. And this could occur experimentally in a lab situation, or it could occur in nature. But essentially what's going to happen is that you're going to get 2 species that are closely related. So let's write that here too. Usually it's not 2 distantly related species. Usually they are closely related. Okay, that's all I have for this podcast now. So let's close with a quick summary. In this podcast, we talked about, in general, how do we make aneuplooids or polyploids? And we talked about non-disjunction. And how that was really effective at making trisomy individuals or monosomy individuals. And this typically occurs in meiosis 1 or 2, but then we talked about a special case where non-disjunction occurred during mitosis in that early developing embryo. So that led to what we call a mosaic phenotype, where some of the cells had the trisomy or monosomy, but most cells don't. And that allows certain individuals to survive longer than their counterparts that are completely trisomy 18, 13, 21, or some other chromosome. We also talked about these 2 terms that are more likely linked to plants, and that is the differences between autopolyploid and allopolyploid. Okay, that's all I have to say for this podcast. If you have any questions at all, please make sure you come see me. I'll be happy to help. See you in class. Bye.